maybe the lack of attention pointed toward R&D investment allocation is reflective of the rosy picture coming out of the JPM conference a couple of weeks ago. Everyone's optimistic that the money's going to be flowing freely again. Sure. I want to jump into some specific questions around steps in the process, Bob, and I want to turn to you for this. So I'm gonna just rattle through a couple of the specific steps, and I'd like you to reflect on your fundamentals for each of these, and perhaps some thoughts on how efficiencies are being improved and can continue to be improved along the way. So the first one I want to throw at you is biological assays. Maybe walk us through sort of the fundamentals and thoughts on where there's room for efficiency improvement.
Yeah, I think that there's, this starts very early on in terms of how you want to build out your assay system. Now, it could be an enzyme, and then you're taking it out as it's alone in a tube, and then thinking about that, you really want to make sure that you actually have the, moving from that into a primary cellular type of environment where you actually can have all the gemisch present that can drive, the response that you get. And there's clear examples of that in like the kinase field where, people have looked for kinase inhibitors and they move them into cellular systems and they don't really replicate the way that they thought they would. So there's a lot riding on this in terms of making decisions of how things respond. And I think being as true to the biology as possible, and this could be organoids and other things where you really are trying to figure out, that biology and how it relates to the disease state, to me is really the important thing. So as we think about this artificial intelligence and we're generating more proteins, more small molecules to make decisions on, and you're using using virtual ways of doing that, you still have to move into the test tube to really figure these things out, and then eventually into humans, right? So you want to make sure you trust, but then verify the activity that you need. And in my experience, starting at least with the primary cell is the most important thing to look at first.
Mm-hmm. Okay. The second one is, and we've spent quite a bit of time talking about this, developability assessments. We've talked about the criticality of developability assessments. Can you share with us how to do that in an efficient? I mean, it seems to me as though that could be a very time-consuming process. Obviously, it requires integration, discussion. A bigger team than probably just the team in research. So how do we get through that in an efficient manner?
Yeah, so within Amgen, that's built into the process. So there is the design that happens, and that's through machine learning and artificial intelligence. All those things are put into that, and then you get into the testing period, which is the automation component of things, and then the reiterization of that process. So it's like an SAR piece that goes in there. And that, I think it's going to change over time. I think there's going to be, as we get more data, we're going to understand this more. You may not have to do the number of tests that you need to do currently to get to the data set. And that would be, once again, so you get the virtual components, but then you have to validate it within the biological system.
Okay. All right. Related question just came across from Caridad. Would you consider a process development, e.g. formulation dev, downstream role in a cross-function developability assessment?
Yeah, I think that is, it's actually crucial, especially if you can't get to the molecular design properties where you still have some liabilities. The formulation can really change things quite a bit as you move forward. So that does come in. It tends to be a little bit further downstream. And I think one of the things that will happen is once we get to, this iteration with this artificial intelligence component of things, I think once again, that's going to be a piece that will be part of that. So it will become further into the mainstream as things go along.
Okay. All right, and then the third area I wanted to ask you about, describing fundamentals and sharing some opportunities for efficiency improvements, are CQAs, so critical quality attributes.
So once again, this comes back to the patient, right? It is all about the patient and the patient experience and getting to the disease and knocking down the disease. So really, that's really what all of these attributes come down to. is getting to the level of efficacy that you need, they think you're going to need in the patient, making sure that you can get to the drug-like properties so that you can actually cover the target for the amount of time that you need to drive that. So that's really what it all comes down to. So you look at both that potency and the drug-like properties of the molecule all get built in right from the very beginning.